Hello and welcome to IB Bio 1.5 Notes. So first we'll be talking about the origin of cells. So Louis Pasteur, he disproved the idea of spontaneous generation, which was widely believed at the time, through his experiment. So his experiment involved the use of swan-necked flasks, as we see here, that were filled with nutrient broth, right here, and they were sealed, which allows no access to outside air. And then some of these flasks were boiled to kill the living organisms inside of them, so all of these, and some were left unboiled as a control group. So in the broth that was boiled and sealed, no microorganisms developed after a long period of time, which disproved the idea that they could spontaneously generate spontaneously. So the broth in these flasks was in contact with air sealed into the flask, and this rejected the theory. And then it was also widely believed that cells came from non-living material, which was also proved by the Miller-Urey experiment. So this experiment involved conditions of early Earth. So they were recreated in an enclosed environment. And after a week, simple amino acids and complex oily hydrocarbons were found in the reaction mixture. So this experiment proved that non-living synthesis of simple organic molecules was possible. So organic molecules can be formed from non-living material. So the conditions for the emergence of this life includes, you're gonna have to write these down and remember them. So simple organic molecules such as amino acids, fatty acids, and carbs must be formed. Larger organic molecules such as phospholipids, RNA, and DNA must be assembled from simpler molecules. The replication of these nucleic acids must be possible, and the biochemical reactions require set conditions such as pH. So therefore, self-contained structures such as membranes are necessary. Now, onto the endosymbiotic theory. So, first of all, the first organisms to exist were all prokaryotes and unicellular. And the endosymbiotic theory states that these prokaryotes were engulfed by other cells, which created eukaryotic cells. And instead of being broken down, they remained inside the cell and provided host cells with energy. So evidence for this are mitochondria and chloroplasts, as they both have double membranes, as expected for cells taken in by endocytosis. They have circular naked DNA, as in prokaryotes, and DNA is formed as single chromosomes, also as in prokaryotes, as well as they have 70S ribosomes, as in prokaryotes. So these are organelles inside the eukaryotes. It is believed that these organelles used to be their own prokaryotic cells. They were engulfed by other cells, and this is the evidence for it. They also divide by binary fission, as in prokaryotic cells and not eukaryotic cells. And they are susceptible to some antibiotics, which is not a eukaryotic trait. Okay, that's it. Thank you for watching.